So one of them. So here's the chemistry. This is actually one of the first pentasaccharides to make it to human use. This is used against thrombosis because there are other aspects besides cancer, which you don't have to go into. But this is a pentasaccharide. This is an inhibitor, a small oligosaccharide inhibitor, heparinase. Heparinase will come along and it will cleave this pentasaccharide into a disaccharide and a trisaccharide. There's a double bond somewhere which I'm missing, and a development of an analytical technique showed a colorimetric assay for this cleavage, and you can measure the cleavage by UV visible. So we decided to examine the cleavage in the presence of our compounds. We decided to come up with the name metalloshielding, hoping that will make us a nice paper and a grant proposal. And so we incubated so what we did was we incubated the pentasaccharide, which we know will bind to our compounds, for 10-15 minutes with a highly positively charged species, and then introduced it to the enzyme. And you see that the control was we measured this as 100% control. This is without the compound. Cisplatinum has no effect on the enzyme, but on the enzyme cleavage, when we start getting into these polynuclear, highly charged species, you see dramatic inhibition of the cleavage of the pentasaccharide. And in fact, these compounds, such as BBr3464 and this 8+ non-covalent compound, are actually more effective than polyarginine. So what we've done is we've protected the cleavage by laying down a large cation on these pentasaccharides. So we're very excited about this because it raises the possibility that we can actually develop these compounds to be active against heparinase or to prevent heparin sulfate cleavage and release of growth factors. Why is this important? It's important because that biological mechanism leads to metastasis and angiogenesis. And so cisplatin is a cytotoxic drug, and we think we have the possibility of changing a cytotoxic drug to an anti-angiogenic and anti-metastatic drug by, instead of attacking DNA, let's attack the glycans on the surface of a cell. And I can't, since this is public, I can't tell you some of the latest results we've had, but they're very exciting and they sort of confirm our theories. So here would be a tumor cell. Here's the heparinase. These are these growth factors. This is heparin sulfate. The growth factors on the heparin sulfate. Heparinase will come along and cleave. The growth factors will go to the tumor cell and induce metastasis. And then we're going to coat the heparin sulfates with these long cationic polymers containing platinum, and we will either prevent heparinase binding, which we're not quite sure about whether we do that, but certainly we look as if we can prevent growth factor release from the heparin sulfate growth factor, and thereby inhibiting metastasis. So we call this area Metalloglycomics, because we think that we can systematically attack the third major class of biomolecules. RNA, DNA is one class, and proteins, of course, are the second class. The third major class of biomolecules are the glycans and the proteoglycans. And now I think for the first time we're beginning to see how we might systematically develop the understanding of platinum compounds with, I'll answer it later, platinum compounds with glycans. Okay, so that's still an interesting point. This is the membrane. It certainly seems to be the way it gets into cells. So there are two aspects. One, how does the heparin sulfate binding enhance tumor selectivity and uptake? And secondly, what are the functional consequences of heparin sulfate binding? So we've begun to look at drug trafficking from the membranes to DNA, because we started this whole story off by looking at a crystal structure DNA, which is supposed to be the target for platinum. And there are a few non-invasive techniques available for studying platinum drug transport. As I say, use of dyes may affect pharmacokinetics. We've begun to look at X-ray fluorescence. Other similar techniques are useful, but recently, with our collaboration in Australia, we've done some high-resolution quantitative imaging using a SIMS-stable isotope mass spectrometry, where you shine a primary beam onto a sample. This was developed for materials. I think one of the interesting things in the last maybe 10 years is development of translation of materials techniques to biology. To samples has been a dramatic increase, and you can sputter off ions off the surface of any surface, a cell or any material, and you can move this beam along, and so you can localize these ions and localize where the compounds are. So this is the basic idea of the technique. We had, for NMR purposes, we had many 15N labeled compounds because we do a lot of HSQC on DNA. And if you do a 15N labeled compound, then you only look at the nitrogen and the ammonia, the nitrogen and the hydrogen attached to it. And so we can examine kinetics of DNA binding, and we've done an awful lot of that. And so we decided to expand and use these double labeled compounds for nanosins imaging, because now we have two elements we can look at, platinum and also 15N. And this is a MCF7, which is a breast tumor cell treated with triplatinum NC after about an hour. And we see the red dots of the platinum around the membrane. And then this is an overlay of both 12-15N and 12-14N. You can overlay natural 15N and natural 14N on your samples. And you see after 1 hour, you see the platinum entering quite quickly. Cisplatinum, you would not see anything at this point. You can try to localize with phosphorus. You don't see any phosphorus localization because phosphorus will be, much of the phosphorus will be tied up in the nucleus. And so we begin to see the platinum enter the cells with this technique. And after 2 hours, the platinum has moved away from the membrane and actually ends up, this is one of our best slides, this is actually the nucleolus. And this gray arrow shows a high concentration of both 195 platinum and 15 nitrogen actually in the nucleolus of the cell rather than the membrane after only 2 hours. So it was a surprise for us. We expected the nucleus, but in actual fact it looks like these compounds go to the nucleolus and can possibly inhibit transcription directly rather than through DNA binding. So we've been looking at these techniques, and I just wanted to show you them because I think they're pretty pictures. We managed to get a ChemCom cover out of this, which was designed by our students. So you shine in the beam and you knock off platinum at 15N, and you can map distribution and trafficking very nicely in this with these techniques. And we have samples now ready. This was actually a breast cancer cell, but it's difficult, the machine is difficult. You certainly need extreme expertise to be able to work with this technique, but we actually have samples of CHO cells, heparan sulfate knockout cells, and the heparan sulfate chondroitin sulfate knockout cells, so we can actually begin to examine if there are any differences in those three cells as we study the biodistribution of the compound from all the way from the membrane into another target is DNA. So of course this is interesting because it means that we can possibly think of a dual function agent. Some of the drug will get to DNA, but also some of the drug will have these consequences of heparan sulfate protection against cleavage. So we, as I say, are excited about this new area, metalloglycomics. It has many applications: identification of the receptors, a new mechanism of accumulation, potential for tumor selectivity, possible use as a heparinase inhibitor. I'm not quite sure about that. I think it's a growth factor release inhibitor rather than a genuine enzyme inhibitor. And for these highly charged non-covalent compounds, possibly dual mode of action is possible. So this begins to maybe start to explain some of the early clinical results we saw with the BBR3464 versus cisplatin. Is there a metastatic component or an anti-metastatic component to the drug that we had overlooked because we didn't understand this part of the equation? There are, I'm not sure I know how to do this, but as I say, we had one result where one patient survived over 3 years after coming back to the clinic with relapsed ovarian cancer for cisplatin, but only received BBR3464, received no other drug, and survived another 3 years or so, which is a remarkable result. And so those tissues are somewhere in a basement in Hammersmith because the NHS, wherever Darren Dixon might complain about the NHS, they're very good. And so I'm sure these samples are present, and we can maybe possibly begin to reanalyze them and examine what was specific about this patient. Is it somehow related to this new findings that we're coming across? Okay, so I'm going to leave it at that. You're all tired, you're all thirsty. And so I won't talk about the zinc fingers. We can talk about that some other time. I'll be talking about that tomorrow anyway in my course. Needless to say, acknowledge the people who do all the work while I go to Brazil. And Alex Hegmans particularly, because he was the one who came up with the 8+ compound. Lauren Williams did the crystallography. Evelina Silva was the first person to show the receptor mechanism. And without Erika Peterson, my lab manager in my biology lab, I would not be able to do any of this stuff. And NIH, NSF, American Chemical Society, touch wood. And I have greatly benefited in the last few years from Brazilian students through CAPS, FAPESP, CMPK, for Evelina and others. And we can all have a bottle of Cisplatino afterwards if you want to tell me how you can have a bottle of wine called Cisplatino. Anybody know? This is a serious question. Anybody know why you would have a bottle of wine called Cisplatino? It's horrible wine, by the way, but anyway. Anybody know? Huh? No. It's Uruguayan. Does that give you a hint? No, I know this story. No Brazilian knows this story. Brazil occupied Uruguay. Not when Uruguay beat you in the World Cup, but beforehand. In 1830, after the breakdown of the Napoleon Empire, breakdown of the Spanish Empire, Brazil occupied the province of Uruguay, which was originally ruled by Buenos Aires, and they called it Cisplatino, this side of the River Plate. Argentina was Transplatino because it's the other side of the River Plate. And so the Uruguayans make this wine called Cisplatino in memory of this. It was about 10 years or so, and then there was some sort of treaty, and it became independent. But anyway, I'll just leave it at this. I thank you all for your attention. Once again, thank you for the invitation to be here, and that's it. Thank you.
It's simple for your talk. I have one simple question for you. You have a very nice compound. So, which one you believe that you will be in the market in the next few years?
Well, that's a good question because obviously, we got to phase 2 clinical trials with BBR3464, which is a big deal. We're the only academic group to ever get a drug into the clinic. And so you could think about modifying it. You could change the chloride to a bromide. You could change the backbone in some way or make it more sterically hindered. But BBR3464 was obviously a paradigm leap from cisplatin. You didn't need a neutral compound. You didn't need 2 chlorides on the same platinum. So we looked over the years, spent a lot of time actually looking at making more stable BBR3464s. But the next real big change is this non-covalent compound. I'm really keen on this non-covalent compound because it's got it'll have a different toxicity profile. It doesn't react with glutathione because there's no chloride to displace it. And it has these very, very strong membrane heparan sulfate effects, which are charge related. So about 18 months ago, we were playing around with both, looking at analogs of BBR3464, see if we could get a better one. But that would only be an incremental increase. And the therapeutic index of all of these compounds is rather low, which is another problem because they're very potent, they're nanomolar cytotoxic. And so, we were seeing therapeutic indices of 1 or 2, which isn't good. Cisplatin is about 6. So I've actually now come around to decide that our lead compound is this 8+ compound. It's totally different. It's clearly got strong membrane interactions, which much stronger than BBR3464. So it's the next, and it also is the next change. You do not need covalent binding to develop a platinum anti-cancer agent. We can measure apoptosis with these compounds and do all that sort of stuff. So we've actually designated this 8+ compound. And so we're looking at anti-angiogenesis versus cytotoxicity, And there's a balance, because if it's anti-angiogenic, you don't want it to be too cytotoxic. That's not a good thing. You have to get a balance. But we've decided that we're going to push for these non-covalent compounds as the most likely to do something totally different. And you sort of need that, because being a metal, and even though platinum is very useful, the barrier is always higher for a metal-based drug than it is for kinase inhibitor, you know, or anything. I got crazy going to the American Association for Cancer Research. There are rows and rows and rows of kinase inhibitors. It drives me nuts. But the barrier is higher, so I think this is different. We've decided that these non-covalent compounds hold the most promise. And also, they represent an interesting tactic versus the oligosaccharide story, because if you look at most of the ways of inhibiting heparinase, the sugar chemists make these extremely complicated tetra, penta, hexasaccharides, which are the recognition sequences of the heparinase. And it just seems to me incredibly complicated to get these things right. So they're using a negative charge to block the enzyme. We're saying, why not use a positive charge just to coat the heparin sulfate? So that's where we're going. Yeah.
You demonstrated that polysaccharides have a role in the transport of that.
Yeah.
What's exactly the mechanism? Is that endocytic?
Yeah, it's an endocytic. Yeah. Which gets me off the hook every time. But it is. It seems to be an endocyte. But that's, people ask that question. Okay, so what happens next? Where does it go? But it's an endocytic mechanism. Yeah, into the cell.